Welcome, everyone. Happy to be here. Happy that you joined us virtually. I'll tell you a little bit about this seminar, asking questions in biology discovery versus knowledge. So what is motivating this seminar? I'll start with that. I've had discussions with many of you who might be joining us virtually because I led this seminar back in 2012, I think it was, which is surprising to me. It was that long ago. But sadly, in many ways, the topic is still relevant in ways that I'll get to. But it's motivated by discussions with especially middle school and high school teachers who had told me how difficult it is to engage in discussion in the classroom, that students were very reluctant to chime in. And in short, this is probably revealing their problems with showing ignorance to either their peers or to their teacher and their instructor. So that's too bad because, of course, we want to engage in these discussions and really dig into the material, often in the classroom. So this is the same observation that I see in the classroom at Yale. And when we started talking about this, K through 12 teachers and me, this shocked them that I would have so many problems also in my classrooms, getting students to chime in and contribute to the discussion. So overall, that is really what is seen in elementary school classrooms. As many of you know who are listening, they just can't stop asking questions. They're super curious. They want to know answers. They are not fearful of sounding ignorant or wrong because they're just curious and they're happy to show it. So something has happened along the way, obviously. There's a stifling of curiosity that I think we should be concerned about in the classroom. And the question is, what could one do about it? So we want to, that's the overarching motivation behind it. And I'll tell you, as a biologist, what is true for me and other scientists is that I'm very comfortable being wrong. That is because the act of science is to ask questions that are either supported or refuted. We call this hypothesis testing. And many times we infer incorrectly, and we learn more often when we are incorrect, frankly. When we're wrong, it helps open up a whole suite of new questions that helps us probe why did we think the prior in order to lead to what we observed in the latter, and how can we bridge between this and ask new questions to bridge to what we call between discovery, is these little bits of things that we learn along the way, to ultimately the confidence in something that we would call knowledge. So this is the bridge that I want to talk about. And I'm, of course, looking at it through the lens of biology, but just to start with this little intro and to end with it, I guess, is the intro is that the lay public just doesn't really understand how science is conducted. And I think that is on scientists. We've not communicated that very well because it is not about being right or wrong. It is about asking questions properly and looking for the evidence to support them. And I am very happily working in a profession where years from now, better tools will be available. And for all I know, every last thing that I claimed will be overturned. And I'm totally sleeping well at night knowing this. So I think that this is a mismatch between what people actually do in the science profession versus what the lay public understands to be science writ large. Okay, so in the process of looking at this issue, there are many things that we're going to dig into using articles, book chapters, probably some discussions with other experts who join us briefly virtually. We did that last time, and I think that that could be a very fun thing. I'll figure out a way to bring reggaeton in and maybe Bad Bunny. I don't know, but I'll figure out a way to do it. So, what does it mean to examine a biological question in the first place? And specifically, how do we formulate a hypothesis that can be supported or rejected? That deserves our attention, at least initially. We're going to look at famous scientists who are famous and, deservedly so, but by no means did they get everything right. And these are people who are working outside of biology historically. Albert Einstein, for example, within biology. Just this morning, I was presenting a lecture to undergraduates talking about Charles Darwin. It was an evolutionary biology course, and I was explaining to them that amazing insights can come from very limited understanding of fields. And Darwin got a lot right, but it wasn't everything that he got right. And the point is, he had no clue what a gene was. And this kind of mystified the students. Like, what? How did he not know what a gene was? Well, actually, nobody knew what a gene was at the time when he was talking about his most famous ideas. And later on, as we fill in the gaps in our knowledge, then we come to understand even old ideas and look at them through a new lens. But all of this is like updated information constantly through time, and we need to live with it this way. So we're going to look at scientists who so-called got things right versus wrong, and yet are very well regarded through time. We're also going to look at scientists who are marginalized, because part of this issue is who has contributed to science historically and who has not. So it has usually been something that the elite or the upper class does, and very many people were marginalized, especially women, people of color through time. So that has led to amazing people contributing, like Rosalind Franklin and Ernest Everett Just, people who were not able to achieve things in their careers because they were just not of the right background to be thought to be good contributors to the science profession. So getting back to the kinds of topics that we'll look at, I want to examine especially some biomedical issues that are probably known to you, the emergence of diseases. Back when we were worried about a new strain of flu, H1N1, I always look back in time, I think it was the early 2000s, where this was all over the news. We've got a new strain of flu, everybody should be worried, and then it kind of petered out because it was not as dangerous as everyone thought, including the experts. So this kind of explanation to the lay public is difficult. It sounds as if we are just flat wrong in our predictions, whereas we make a prediction, and if we are predicting that the variant of flu is going to be dangerous or not, we have to live with that prediction because we're producing vaccines alongside it in time to reach the flu season. And we may have a mismatch. So this is just not a perfect science, so to speak. And this is an example of how well-known things like catching the flu, of which we're very worried about right now in the cold and flu season, how it's very difficult to predict what is going to happen. And of course, we'll dive into epidemics and pandemics, especially the one that you've lived through, as looking through the lens of exactly how did that start? What is the controversy behind it? What is the enduring controversy behind it that is now a political issue that affects our lives in terms of hygiene practices, wearing masks, and other things that we're either on board with or not, and how it's become a politicized issue. Let's see, what else we'll talk about are infamous things that have occurred in the name of science, eugenics, the Tuskegee syphilis experiment, where people were denied treatment for a deadly disease knowingly. And this was well after the Nazi doctors were taken to trial and said that this is a very wrong practice. We can never let this happen in the world again, and yet it happened on US soil. So we need to dig into this and look at it. The very famous case of where new medicines come from and new discoveries that lead to knowledge come from. Sometimes it's on the backs of materials that are taken from people who are marginalized. And the great example of that is Henrietta Lacks and the very famous HeLa cells that, unfortunately for her and her family, originally, well, she died in the process of having cancer that was aggressive, but her cancer cells were isolated and used at Johns Hopkins, spread around the world, including off Earth on space station, where experiments occurred there, and her family never got a dime until this was fortunately revealed by an expert journalist in a very well-written book. I don't recommend the movie because it's not so good. So we're going to look at these various things through time that have kind of pushed at the lay public's faith in the scientific enterprise. And I feel like it's all wrapped up in how do you address scientific questions properly, ethically, and what does it really mean to be correct or not when you're making scientific discoveries that ultimately lead to knowledge. I think that the best case scenario is for teachers who are in biology, the life sciences. They're going to be most comfortable here. Usually for my seminars, those teaching in mathematics, we can find some good common ground because of the mathematical biology that can go along with it. This might be a particularly difficult seminar for those who are outside of those fields, not because they can't handle the material, it's because of the material on the side to understand the terminology and everything to keep up with it. That might be a little too foreign for you to make good progress in a very abbreviated 2-week period. Other than that, I think I've held forth long enough. So thank you for your attention and appreciate your time in listening.